Okay, so on to the actual content. So we're going to start the lecture just talking a little bit about signal transduction. At this point, you should be familiar with what it is, basically, what its function is, how it works. So I put this graphic here not to stress you out, not to make it seem like there are 50,000 things that you have to remember. Just you have to keep in mind that when we're talking about signal transduction, we're talking about a bunch of things going on all at once. So there are hundreds, potentially thousands of signals that are being transmitted through the cell at any given second, and a lot of times these signals have multiple targets. So it's very complex and it's very delicate. So that's basically what I wanted to just summarize with this picture. But in no way do you need to remember these names, anything like that. You just have to understand the general flow. And we're going to talk about that right now. Starting at the top, you have those surface receptors. So what is signal transduction? Cells can communicate with the outside world via surface receptors. So when the extracellular domain of a surface receptor is activated, the receptor will effect a change inside the cell. So if we look right up here, these are two proteins. They're both most likely receptor proteins. And you could see this top over here where both of these proteins have these sort of two heads sticking out. That is the extracellular domain, because that is where the receptor is actually reaching outside of the cell to communicate with the extracellular environment. So that's if we've spoken about receptors before, we talk about induced fit model or the lock and key model. This is the lock part. So the key would actually be the ligand, or that cell that's going to come and sit right on top of the receptor and start the signal cascade inside. But that specific part that the ligand binds to, we refer to as the extracellular domain, and that's the part that's actually sticking outside of the cell. So receptors may be activated by many different types of signaling molecules. These signaling molecules are also known as ligands, L-I-G-A-N-D, as I was just referring to it before, same thing. But they can be hormones, neurotransmitters, they can be cytokines. It really depends on what the pathway is and what the target is. And some receptors don't even have an extracellular domain, and they're located deep within the cell. So for example, when we talk about the different types of hormones, one of them, these steroid hormones can diffuse through the cellular membrane. So a lot of times they have these receptors that are deep within the cell, like estrogen, for example. It works very deep inside the cell. It's not a surface receptor, but it still does, result in many of the same things as these do. I mean, there's a signal cascade that follows. There's signal amplification, and then it affects the entirety of the cell. So the job of a surface receptor is to transmit information from the outside of the cell to its internal environment without ever letting the signal molecule pass through the cell membrane. So in most of these surface receptors, nothing is actually passing through the cell. So the receptor will utilize second messengers inside the cell to carry out the signal cascade started by the original ligand. So that's why we have these secondary messengers. The ligand, if you look over here in this diagram, the ligand are these little orange pieces. That they're also referred to as the primary messenger, because that is where the signal starts. So that is the primary messenger that is going to start the signal cascade. It interacts with the receptor, and then it does not pass through the receptor. The ligand stays on the outside, and when it's finished doing what it has to do, it'll probably just dissociate from the receptor and go back into the extracellular environment, where maybe it'll be degraded or taken up by something else, but it does not actually go into the cell. So because it doesn't go into the cell, that is why we have to utilize these secondary messengers. So those are the ones that actually carry the signal cascade. It's sort of like a relay of information from one molecule to the other. That's basically what the signal cascade is. And at the bottom of the screen, just to define signal cascade, is the series of biochemical reactions that follow the activation of a surface receptor by its ligand. So the point of the signal cascade is to amplify the signal as it proceeds. So if we look at the diagram to the left and we go in order, we have 1, the receptor ligand binding. So that's going to start the signal. 2, signal transduction via the secondary messengers, whatever that secondary messenger may be. We'll talk a little bit about it in a couple of minutes. If it's a G protein, for example, then that receptor is going to mobilize that G protein, and it's going to start its effect throughout the cell, which is 3, the cellular response. And then, oftentimes it is a change in gene expression, because when we're talking about this, and especially when we're talking about, cancer cells later, we're really talking about changes in gene expression that regulate cell proliferation and cell growth. So those are super important things to keep in check. But they're also super important things, to make happen as quickly as possible. So signal transduction allows us to do that. Okay. And we're going to talk a little bit now about cyclic AMP, also known as CAMP, and the enzyme known as adenylyl cyclase. So adenylyl cyclase is also known as adenylate cyclase. It's the same thing. It's an example that is very often used, especially when talking about signal transduction. And it will probably pop up on the MCAT on some sort of section. If it's not in the biology section, then maybe it'll pop up in the psych-soc section in something that you're reading. So it's a really good idea to be familiar with this. You definitely should understand it. So adenylyl cyclase is an enzyme which converts ATP into cyclic AMP, which is also known as CAMP. So when adenylyl cyclase is activated by its G protein coupled receptor, the second messenger CAMP then carries on the signal. So G protein coupled receptor is a type of surface receptor. If you look right over here at the diagram at the bottom, and we start all the way over to the left, this sort of oblong blue is the G protein coupled receptor. And then you can see 3 smaller purple things attached to the bottom right. Those are the 3 subunits of the singular G protein. So the G protein is broken down into alpha, beta, and gamma. All of them have their own rules. But the important thing to know is that when that G protein coupled receptor is activated by its ligand, that's going to activate the G protein. So the G protein is going to dissociate. It's going to exchange GDP for GTP. It's going to become active. And then it's going to activate adenylyl cyclase. And then adenylyl cyclase is going to turn ATP into CAMP, cyclic AMP. So you could see how it's very sequential. And then once cyclic AMP is made, the levels of CAMP in the cell are going to rise. It's going to activate protein kinase A, and it's just going to continue the signal cascade. So it's like just a very sequential, step-by-step process when we're talking it out like this. But like I mentioned before, keep in mind that there, thousands in a given second are going on, and your cell is doing all different kinds at all different times. So there are a bunch of weird interactions that, in vivo actually do happen. So, right, pKa, which is protein kinase A, will then phosphorylate and affect the other target cells, and it's going to contribute to the metabolism of glycogen in the cell. So pKa is actually activated when we're trying to mobilize our glycogen stores, our fat stores for a fight or flight response. Okay, so I'm going to put a review question in here for you guys. I would take maybe no more than a minute to answer this. I'm not going to wait a minute, so just pause, take your time, and when you're done, you can unpause. Okay. So which of the following is not true regarding signal cascades? So the answer to this question is going to be D, and let's go over it. So A, the signal originating from one receptor will be amplified many times over as it travels down its pathway. So the question is asking which of the following is not true, and that is 100% true. That is the whole point of the signal cascade, is that it's amplifying the message. So it's a really effective way to transmit information throughout the cell. So A is correct. A is a true statement, so it is not the correct answer. Because remember, again, we're looking for what's not true. So B, some signals may be first received by receptors deep within the cell. Well, we did speak a little bit ago about how estrogen works like that. It goes deep into the cell because it's a steroid hormone, so it can actually pass through the cell membrane just by simple diffusion. So some signals do actually start with receptors deep inside the cell. So B is also a true statement, so it is not the correct answer. C, cyclic AMP, also known as CAMP, is a common second messenger in humans, which it definitely is. We just talked about the CAMP pathway. Your cell uses CAMP levels to adjust pKa activity. So like we said, if CAMP is upregulated, your cell is going to upregulate pKa, and we're going to mobilize those glycogen and those fat storages for the fight or flight response that we have. So yeah, so C is a true statement, which makes it not correct for this question. And then we finally get to our correct answer choice D. The strength of the signal diminishes as it is transduced throughout the cell. That's not true. It's actually directly opposite of what A is. So if you knew that one of them was, if you could identify that they're sort of antonyms, then you would be able to narrow it down to just A or D. And hopefully you would remember that signal amplification is a part of signal transduction. So the signal actually gets stronger. So D is not true. So that makes D the correct answer.